Good afternoon. This is the first session about heredity, and we are going to start by defining heredity. Heredity is a science, and it is the science that studies the transfer of genetic information from parents to offspring. Let's move on to the first slide on that presentation, which is meiosis. As you know, we have two types of cell division. The first type of cell division was mitosis, and mitosis was the one we studied during our study of the cell cycle. In mitosis, you start by a parent cell. Let's say that the parent cell is 2N, and then you do one shot of nuclear division followed by cytokinesis or cytoplasmic division in order to get rise to two daughter cells, and these two daughter cells are exactly identical to the parent cell just because they have the same genetic makeup. They have the same number of chromosomes and the same type of chromosomes. This type of cell division, as we have studied in Unit 4, is important for growth and for repair of wounds. But this type of cell division we are studying today, which is the second type of cell division, is meiosis. And meiosis is mainly done by living cell in order to produce gametes. Gametes have another name, and gametes are also called reproductive cells, just because the fusion of gametes is an essential process in sexual reproduction. And that's why usually gametes are haploid, meaning that they have half the number of chromosomes. What's the importance of haploidy of gametes? Now, if we consider a haploid sperm having half the number of chromosomes, which is N, and an egg or an ovum having, again, the haploid number of chromosomes, then the fusion between the two will result in a diploid zygote. Thus, we are preserving the chromosomal number of the species. If meiosis does not involve reduction of the chromosomal number or dividing the chromosomal number by 2, then for every fertilization, there would be multiplication or duplication of the chromosomal numbers. The chromosomal number. I'll stop sharing for a while to make sure I'm sharing the proper slide. Yes. Okay. So meiosis is important because it is the process by which we divide the chromosomal number by 2 and we obtain a monoploid or haploid. Haploid means, when I say, for example, a haploid gamete, I mean a gamete or a reproductive cell that contains half the original number of chromosomes. And since the original number of chromosomes in most organisms is diploid, then a haploid gamete will contain only one N or one set of chromosomes. And that's why haploid in diploid organisms may be equivalent to monoploid. Okay? So meiosis is called reductional division because when meiosis is complete, it results in halving or diminishing the number of chromosomes by dividing it into 2. Actually, mitosis occurs in one shot, but meiosis occurs in 2 shots. So if I start by a germ cell that will be converted or will divide by meiosis to give rise to a female reproductive cell or a male reproductive cell, that then it will undergo two shots of nuclear division or meiosis. The first one is called meiosis 1, and it is the part or the stage of meiosis in which at the end of which we really get haploid cells, or I mean that actual division of the chromosomal number occurs in meiosis 1. Meiosis 1 has the same phases as mitosis, but we add the Roman number 1 after each phase. So we say that meiosis 1, for example, consists of prophase 1, and then metaphase 1, and then anaphase 1, and then telophase 1. And then following meiosis 1, we have another shot of nuclear division in which we divide each of the already present daughter cells into two more daughter cells, but this time they having the same number of chromosomes, and this second stage is called meiosis 2. In meiosis 2, we start by N, and we end up by having two daughter cells, each having N. So we start by a mother cell, and we end by a daughter cell, or two actually daughter cells that have the same number of chromosomes. So we can say that meiosis 2 is similar to mitosis. Why? Because the chromosomal number is not changed or not altered. Okay. Similarly, just like in meiosis 1, meiosis 2 consists of the four actual phases, but this time we would be calling them two. So meiosis 2 consists of prophase 2, metaphase 2, anaphase 2, and telophase 2. And at the end of telophase 2, we will obtain four daughter cells, and these four daughter cells are the final gametes. In case of the male gametes, the four daughter cells are preserved, whereas in the female gametes, only one stays and the rest of them are degenerated because they have less cytoplasm. Meiosis involves diversity of the gametes, meaning that if we consider the four produced gametes, they will not be similar. And this is another difference between meiosis and mitosis. In mitosis, you start by 2N, and you end up by having two cells, each having 2N, and that are whose chromosomes are exactly similar to the original chromosomes. But in case of meiosis, the four daughter cells are different, and this results in diversity of living organisms, because each and every one of these daughter cells, when they survive, will be responsible for making half the chromosomal number of the resulting zygote that will be an individual at the end of the day. Is diversity important? Yes, diversity is important because diversity results in evolution. What is evolution and how diversity leads to evolution? This is what we are gonna learn in evolution, in the unit on evolution, but let's have a very rapid idea about it now. Now, when, for example, say that we have four organisms, 1, 2, 3, and 4, and that those four organisms differ from each other, and suppose that these four organisms are living in a certain environment, and that this specific environment suits the four organisms. and they are all well adapted to that organism. And then there happens to be a sort of environmental stress. There are environmental changes, like, for example, drought or extremes of temperature. And upon occurrence of these environmental stressful conditions, we find out that the first three organisms can cope with the environmental stresses and that they are well adapted, whereas the organism number 4 is not well adapted and cannot survive. And therefore, organism number 4 will, all organisms, of course, of this type 4 will die and pass away. They'll not be able to survive. And if they are not able to survive, then they will not be able to reproduce. Whereas the organisms of the first three categories will survive. And since they survive, they will reproduce and they will have viable offspring, and they will stay representing their variety. And therefore, gradually as time passes, there are changes that occur in the species. If we consider that those 4 variants are variants of the same species, so at the beginning, before the occurrence of the environmental stress, all the 4 representatives, all the 4 variants were present. And then after the environmental stress, one of them has disappeared. So gradually the species would not contain that variant. Of course, the examples are many, but what happened is survival of the fittest. If we want to describe what happened in this example, it is survival of the fittest. And here, the fittest were the first 3 variants. And why were they considered the fittest? Because they were the best adapted for the environmental changes, whereas number 4, being less adapted to the environmental change, could not survive, could not reproduce, and was no more represented in the species. Okay, so diversity is one important factor of survival of the fittest, and this is one kind of natural selection, because this stress has occurred, selected for and selected against. So we can say that this stress that occurred selected for variants number 1, 2, and 3, and selected against number 4. So survival of the fittest is a result of natural selection. A natural selection is the cause for evolution, and evolution is characterized by 3 characteristics. The first one is that, first of all, it is a change, okay? Evolution is a change, but a change that has 3 important specifications. The first specification is that it is a gradual change that occurs over time. The second specification is that it's cumulative, meaning that at the end of a certain period of time, you have a big change, but that big change is nothing but the resultant of accumulation of small changes. And the third characteristic of the change that occurs in evolution is that it occurs at the level of the species. So we can talk about evolution of a species, but we cannot talk about evolution of an individual organism, okay? So meiosis is a type of cell division that allows diversity. But how does meiosis do this? There are several processes that occur in meiosis that help the produced gametes to be diverse. And one of them is mentioned here, which is that the genetic material of the chromosomes is randomly separated and recombined, meaning that the chromosomes in meiosis 1 are present in pairs, and these pairs are aligned next to each other, like these two pens, for example. Let this pen be the maternal pen and that black pen be the paternal pen, the paternal chromosome. Then during telophase and then during cytokinesis, they will be separated into two daughter cells. So say, for example, we are making sperms. So this sperm will contain the blue chromosome, and that sperm will contain the black chromosome. So this sperm will result in the formation of an individual, and that sperm will fertilize an ovum and result in the formation of a second individual, okay? Before separation, they might be like this, arranged like this, or they might be arranged like that, with the black pen on this side. And in such a case, the makeup of this organ, the newly formed organism, will be very different from the first example. So random assortment means random distribution. Will this black pen will be on this side or on that side? Will it go to this gamete or to that gamete? And this will finally result in a different genetic makeup. Of course, we'll be seeing this on the diagram. So there is random assortment or random distribution that will result in variation. There are other causes of variation, of course, that we will be discussing on the next slides. The essential idea in meiosis, or one of the major differences between meiosis and mitosis, is what we see here on the slide. If you look here at the nucleus, which represents prophase of prophase 1 or prophase of meiosis 1, you will find that the paternal chromosome and the maternal chromosome are arranged in pairs. Each of these pairs is called a homologous pair. A homologous pair. What do we mean by a homologous pair? A homologous pair is a homologous pair of chromosomes, actually. And that homologous pair of chromosomes, like this one, for example, consists of two chromosomes. And those two chromosomes are duplicated chromosomes, meaning that each of these two chromosomes is made of two sister chromatids. But the first duplicated chromosome is a paternal chromosome, like this one, for example, and the second one is a maternal chromosome. So every two homologous chromosomes come close to each other and form a homologous pair of chromosomes. And in a homologous pair of chromosomes, every chromosome, the maternal one and the paternal one, is made of two sister chromatids. Why do these two chromosomes in particular, or why do every two chromosomes come in close contact with each other, or what determines this? What determines this is them being homologous. So what do we mean by homologous chromosomes? Homologous chromosomes means that they carry the same genes, or they carry loci. They have loci. Loci is the plural of locus, and a locus is a site or a place. So say, for example, this locus on the paternal chromosome is for eye color, then this locus on the maternal chromosome, on the homologous maternal chromosome, will be for eye color. And if that locus, for example, is for eye shape, then that locus on the homologous maternal chromosome will be for eye shape. Then they carry genes for homologous characteristics. And this doesn't mean, by the way, that both have to be dominant. For example, if the dominant color for eyes is the brown color and the recessive one is the blue one, so the homologous loci that carry genes for eye color should not be necessarily both dominant maternal and paternal, or both recessive maternal and paternal. These homologous chromosomes, when they are put in that arrangement, we call the process that allows them to come in contact with or very close to each other. We call this process synapsis. So synapsis is the process by which two homologous chromosomes, one paternal and one maternal, come close to each other and are arranged next to each other in prophase of meiosis 1. And in order for this to be accomplished, there should be a complex arrangement of proteins, as you see in the middle. So if the blue chromosome is the paternal one and the pink or purple chromosome is the maternal one, then the molecules in between are a complex group of protein molecules that are called the synaptonemal complex. A complex of protein molecules that helps synapses, and that's why we call it a synaptonemal complex. So the process in which a homologous pair of chromosomes are arranged next to each other precisely is called synapsis. And the shape you see is a tetrad. So here, what you see is a tetrad, Y tetrad, because originally every one of these chromosomes is duplicated. So we have 1, 2, 3, 4 chromatids. 1 and 2 are sister paternal chromatids. 3 and 4 are sister maternal chromatids, okay? And it is this process of synapsis that allows you to see the tetrad. This arrangement that you see allows exchange of material between the inner chromatids. So we have exchange of material between that chromatid, the inner blue one and the inner pink one. And this exchange of material process is called crossing over. Crossing over, okay? So it is called crossing over. There is something wrong. Okay, so what is crossing over? Crossing over is this process of exchange of material between the two inner chromatids, as we will be seeing here. So you can see here a tetrad, and you can see that there is this exchange of material. So the blue chromosome gives part of its genetic material to the yellow one, and in exchange, the yellow one gives the blue one the same part, the homologous part. So if this is J, then this would be J. This is a gene, K, K, L, L. And this, although, for example, as you see here, L is recessive, whereas L here is dominant, this doesn't make any difference. They should only be genes controlling the same characteristics. I shape in front of I shape, or in exchange for I shape, eye color for eye color, height for height, and that's it. So this process that occurred is of exchange of genetic material is called crossing over. But the shape you see is a chiasma. So this X shape you see is called a chiasma. So again, we have a process and a shape. How many processes have we seen, and how many shapes have we seen? We have two processes. We have synapsis, and synapsis allows crossing over. Both are processes. Synapsis is a process and crossing over is a process. When synapsis occurs, you see a shape, which is the tetrad. And when crossing over occurs, you see a shape, which is this X-shaped structure, which is called chiasma. Of course, this results in exchange of this crossing over, and this chiasma results in exchange of genetic information, so that every chromosome, because these two chromosomes, at the end of the day, these two whole chromosomes will be located at the end of meiosis 1, they will separate from each other. And at the end of meiosis 1, during telophase and then during cytokinesis, they will be lodged into different gametes. And therefore, this gamete has taken a chromosome that is mostly from the mom, but partly from the dad. Here, on the other hand, the other gamete, say for example, this is gamete 1 or daughter cell 1 and daughter cell 2. Daughter cell 2 at the end of meiosis 1 has received a chromosome that is mostly containing genetic material from the dad with very little genetic material from the mom. And this allows you to be a blend of the characteristics of your mom and dad. But maybe in some characteristics, you're more like your mom. If you take that chromosome, by the way, we call that chromosome a recombinant, or any chromosome that consists of a maternal part and a paternal part is called a recombinant chromosome. So the yellow chromosome here is a recombinant chromosome because it has taken part from the mom, and the blue chromosome is also a recombinant chromosome because it has taken part from the dad. And actually, any DNA molecule that is made of DNA from two different sources is called recombinant DNA. So recombinant DNA, whether it is a piece of a chromosome or a whole chromosome, whether it is a piece of a molecule or a whole molecule, since it contains DNA fragments from two different sources, then we call it recombinant DNA. If you receive the gamete, if you are made of the gamete that contains the blue chromosome, this one, gamete number 1, then for the characteristics, only for the characteristics on that chromosome, you will be more like your mom. For example, if that chromosome carries eye shape, eye color and length of the eyelashes, for example, then the shape of your eyes, the color of your eyes, your eyelashes would be more like your mom. But maybe, for example, the shape of the eyelid around the eye and other characteristics would be like your dad. So it all depends upon which chromosome are you receiving from your dad and which chromosome are you receiving from your it all depends upon which chromosome you are receiving from your dad and which chromosome you are receiving from your mom. Maybe in another chromosome that is also recombinant, we have 23 pairs of chromosomes, maybe in another chromosome, most of the genetic material would come from your dad. So you will find yourself in certain characteristics more like your dad and in other characteristics more like your mom. Okay. Now let's move on. Here again, you can see two homologous chromosomes, the brown one and the blue one, and then the shape you see here, the X shape you see here is the chiasma, but the process, for example, if I ask you about the process here, the process that occurred here is a process of crossing over, and finally you obtain recombinant chromatids. And when we say or recombinant, actually the inner chromatids are here the ones that are recombinant. Recombinant means that they contain DNA from different sources. Here we have two different sources, so they contain DNA from two different sources. But since the inner chromatids are recombinant, then you can consider that the whole chromosome is recombinant. By the way, those inner chromatids, so again, crossing over occurs between the inner chromatids. Those chromatids at which crossing over occurs are called non-sister chromatids. Why are they called non-sister chromatids? because one belongs to the mom and the other belongs to the dad, and therefore they do not necessarily carry the same alleles. They have to carry homologous genes, eye color for eye color, eye shape for eye shape, eyelash length and shape for eyelash length and shape, but not necessarily the same alleles, meaning that they are not necessarily both recessive or both dominant. But what are sister chromatids then? Sister chromatids are the chromatids that belong to the same chromosome. So those two maternal chromatids are considered sister chromatids, and those two paternal chromatids are considered sister chromatids, okay? As you see in metaphase 1 here, metaphase means that the chromosomes will be aligned along the equator of the cell. One important difference is that here, between, of course, metaphase 1 and metaphase 1 is metaphase of meiosis 1. And when I say metaphase only, just metaphase, I mean metaphase of mitosis. One important difference is that the chromosomes are arranged double file, okay, along the equator. And the second important difference is that if you look at the chromosomes under the electron microscope, you will find that the chromosomes are recombinant. Why are they recombinant, or what does this recombinant appearance indicate? It indicates that crossing over has occurred earlier in prophase 1 or in prophase of meiosis 1. Okay, and then we move on to anaphase 1, and in anaphase 1, as you see, the chromosomes move towards the poles of the cell. And again, one important difference between anaphase 1 and between anaphase, which is anaphase of mitosis, is that what is moving here towards this pole and that pole are whole chromosomes. And when I say a whole chromosome, I mean a duplicated chromosome. So meiosis 1 is characterized by separation in anaphase 1 of whole chromosomes. Separation in anaphase 1 of whole chromosomes, and the whole chromosomes are duplicated chromosomes. Remember that we have started by a chromosomal number of 4, and up till now the cell contains 4 chromosomes because we are still in anaphase 1. In telophase 1, there will be separation of the two cells. Cytokinesis will start, and this occurs by a cytoplasmic constriction in animal cells. The nuclear membrane will reappear, the nucleolus will reappear, and now we have separated the chromosomes, meaning that you will have 2 chromosomes in every daughter cell, but they are recombinant chromosomes. So here we have 2 chromosomes. So you might have realized that by the end of meiosis 1, reduction of the chromosomal number has occurred. Now each of the 2 daughter cells contains half the original number of chromosome. Not only this, but something moreover, we have recombinant DNA. And recombinant DNA is important for diversity. If I ever ask you about the importance of recombinant DNA, it is important for variation or diversity. And if I ever ask you about the importance of haploidy, you would say that haploidy is important for preservation of the chromosomal number of the species. Preservation of the chromosomal number of the species. Meiosis 2 is like mitosis. If we look at this diagram, here at the end of meiosis 1, for example, here we started by 3 pairs of chromosomes or 6 chromosomes, and at the end, and here you can see how they are arranged double file in metaphase 1. And at the end of telophase 1 and after cytokinesis, we ended up by having 3 chromosomes only in every daughter cell. So they are haploid, but notice that these chromosomes are duplicated. And because of this, you might have noticed, or you will notice that in interphase, because meiosis 2, just like meiosis 1, will be preceded by interphase, in which there is preparation for cell division. But in interphase, we are used to having the chromosomes or the DNA molecules duplicate themselves. But in interphase 2, which is interphase of meiosis 2, there will be no duplication. Why isn't there duplication? Because at the end of meiosis 1, the chromosomes are already duplicated, so they needn't duplicate themselves. And at the end of meiosis 2, which is very similar to mitosis, we will find that every one of these these haploid daughter cells will form 2 daughter cells, each of them being haploid. But the difference is that here you have duplicated chromosomes, here you have single chromosomes. Why do we say that it is similar to mitosis? First of all, it has the same phases, meaning that we have prophase 2, metaphase 2, anaphase 2, and then telophase 2. The second important thing is that we start by haploid cells and we end up by having haploid cells, meaning that we preserve the chromosomal number, and this is one characteristic of mitosis. But one important difference between mitosis and meiosis 2 is the presence of recombinant chromosomes. We never talk about recombinant chromosomes in case of mitosis. Here there is a useful table that tells you the major differences between meiosis and mitosis. Meiosis involves 2 rounds of nuclear division followed by cytokinesis, whereas mitosis involves only 1 round of nuclear division followed by cytokinesis. In meiosis, we have pairing of the homologous chromosomes that is called synapsis, and that allows the presence of crossing over, and crossing over is one cause of diversity. Crossing over is one cause of diversity or variation between the gametes, and if there is variation in the gametes, then there will definitely be variation in the organisms that are made out of these gametes. Whereas in case of mitosis, there is no synapsis, there is no crossing over. In meiosis, as we said before, in the tetrad, the 4 chromosomes may be arranged, the 2 actually duplicated chromosomes might be arranged like this, or they might be arranged like that. You might ask yourselves, what's the difference? Then we will have 2 daughter cells, each containing one of them. But the fact is that these chromosomes are not alone in the cells. So you might have, for example, for the first pair of chromosome, the paternal to the left and the maternal to the right. But for the second pair of chromosome, which is different, like this, for example, you might have the maternal to the left and the paternal to the right. So for one pair only, it doesn't matter whether they are arranged like this or like that, but for the whole set, it would make a big difference, because you would have different arrangements of the different chromosomes around the equator. Here, the separation of the sister chromatids occurs at the end of mitosis, but here, in order for the sister chromatids to be separated, this occurs at the end of meiosis 2. Here we have 4 haploid daughter cells, Here we have 2 diploid daughter cells. Here there is variation and the 4 daughter cells are genetically different, whereas in mitosis they are similar. Okay. Now we're going to talk about, or to summarize actually, the causes of genetic variation in meiosis. Number 1, as we said, the independent assortment of the chromosomes. For example, here we have, this is what I was just explaining, that if you look at more than one pair of chromosomes at the same time, you can understand why it is important to consider the assortment of the chromosomes. Are they arranged like this, with the black one on this side and the blue one on that side, or are they arranged like this? If you consider one pair only, it wouldn't make any difference. But if you consider a set of chromosomes, it would make a difference. Why? Let's look, for example, at this cell. Here we have 2 pairs of chromosomes, okay, 4 chromosomes. They might be assorted like this. In such a case, when you get the daughter cells, one daughter cell will be fully paternal, for example, having brown eyes and black hair, and the other one will be fully maternal. Okay, so here we can say that this is fully paternal, meaning that this gamete will only contain properties of the dad, or genes that control, that give, when they are translated, they give the properties of the dad, whereas this one contains genes that are all maternal. But if during, of course, this assortment of chromosomes occurs during metaphase 1 or metaphase of meiosis 1, if they are arranged like this, meaning that the first pair decides to be arranged like this, with the paternal, the red paternal one on the to the left and the blue to the right, and the second pair decides to do the opposite, with the maternal, the blue maternal, to the left and the red paternal to the right, then the daughter cell here, if we consider those 2 daughter cells, 1 and 2, then each and every daughter cell will contain 1 chromosome from the mother and 1 chromosome from the dad, which means that there will be more variation. You are not all your dad or all your mom. So independent assortment of the chromosomes, and this occurs during metaphase 1, is one important cause of diversity. Of course, the second important cause of diversity is recombination that occurs during crossing over, and the third one is random fertilization. What do we mean by random fertilization? When we have in our somatic cells, not in our reproductive cells, when we have in our somatic cells 23 pairs of chromosomes, this means that we have homologous pairs that are 23 in number, because the total number of chromosomes is 46. Without going into mathematical details of permutations and possibilities, you can say that this somatic cell, when it decides to carry out mitosis and produce sperms, there will be 2 to the power 23 different genetic recombinations. Why? Because this is what I was just explaining. You're not only considering 1 pair of chromosomes, but you are considering 23 pairs of chromosomes. And in every pair, you cannot know whether the paternal will be on this side or on that side. So you might have a gamete where all paternal is on one side and all maternal chromosomes are on the other side. You might have, for example, gametes where half of the paternal is on one side and half is on the other side along the equator of the cell. You have an unlimited number of possibilities, which is 2 to the power 23, which is 8.4 million. And the same applies to the female gametes. And therefore, because in case of fertilization, what happens is that you have a sperm that fertilizes an ovum. And for the sperm, you have 8.4 million, means times 10 to the power 6 possibility of genetic makeups. And for the egg, you also have 8.4 million possibility. Then the overall probability of the genetic makeups of the zygotes that are made from fertilization of one of these sperms with one of these eggs is the possibility of the sperm, the possibilities of the sperm, or the probabilities of the sperm multiplied by the probabilities of the egg. What probabilities? The probabilities of the genetic makeup. So you can take 8.4 million pictures of, or you can take pictures of 8.4 million different arrangements of the paternal and maternal chromosomes on each side of the equator in different kinds of sperms. So the overall possibility for the zygote, which necessitates fusion of a male gamete with a female gamete, is the first probability, probability 1, multiplied by probability 2. This is the probability of the zygote. Why? It is the probability of the sperm multiplied by the probability of the ovum, because the zygote is made from a sperm and an ovum. So it is 8.6 multiplied by 10 to the power 6 multiplied by 8.6 multiplied by 10 to the power 6. And this is what's written here. 8.4 and 8.4. 8.4 multiplied by 8.4 multiplied by 10 to the power 12, which is nothing but 10 to the power 6 multiplied by 10 to the power 6. So this indicates you that in addition to crossing over, which is essentially a huge cause for diversity, and in addition to random assortment of the chromosomes, which is another cause of huge diversity, you have a third factor that gives you more and more cumulative diversity, that is random fertilization. You cannot know which sperm will be fertilized by which, or which sperm will fertilize which egg. And now we are going to move to a different entity, which is Mendel's experiments. And of course, you know that Mendel is considered the father of genetics because Mendel was the first one. He was a monk, and he was cultivating pea plants in the garden of a monastery. And Mendel was the first one to carry out genetic experiments and to base the information he delivered, or to base the information on experimental basis. Not only this, but it seems that Mendel was fond of maths and using math in his experimental, in calculating and approaching his experimental results, led him to a quantitative approach, meaning that Mendel was the first one to use math and mathematical results and statistical data and analysis of these statistical data. One of the causes, a third cause of success of Mendel's experiments is that he worked on a huge number of plants, because whenever you're dealing with probabilities, as we're going to see on the following slides, if you consider only a small sample of organisms, your results might not give a valid explanation. But the larger the sample you are working on, the more valid your results will be. Also, Mendel was lucky because when he selected characteristics to work on, he selected clear-cut characteristics, meaning that, for example, he selected the color of the flower, and there were two clear-cut characteristics. For example, there were pink flowers, clearly pink, and white flowers. There were tall plants and very short plants, meaning that there was nothing in between that might be ambiguous. So when he came to count the results and see how many are tall and how many are short, there was no confusion. How many are pink and how many are white, there was no confusion. How many flowers are apical at the top of the stem and how many flowers are axial, like making an angle with the stem, there was no confusion. And moreover, this is not written on the slide, he was also lucky because all the genes he selected, or all the characteristics he studied, were controlled by genes that were located on different chromosomes. Actually, the pea plant has 7 pairs of chromosomes. For example, when he studied the chromosome, when he studied, for example, height of the plant was located on a chromosome. Color of the flower was located on a different chromosome. Therefore, these traits he was studying were unlinked. They were not on the same chromosome, because if they were linked, present on the same chromosome, they were he would have obtained different results. So we can add that number 5, he studied 3 genes, non-linked genes, meaning that the characteristics or the traits he studied were controlled by genes that were free, non-linked, present on different chromosomes. They were present on different chromosomes. Sorry. And now we move on to a new concept, which is probability or likelihood. The probability of something to occur is the likelihood of something to occur, how likely, how possible this event can occur. If we say that an event has a probability of 0, this means that this event cannot happen. Just like, for example, when you're talking about somebody who has passed away like 100 years ago, and the likelihood that this person is nowadays alive is, of course, 0, because he is dead or he has passed away long ago. The other possibility is probability 1, and probability 1 is a likelihood that is certain, that is absolutely certain. And when we say 1, we mean 100%. Like, for example, the probability that I am alive now is 100%. The probability is 1 because I'm actually alive. There is no doubt that I am alive, that I am talking. But probability between the two, between 0 and 1, it's not impossible, and at the same time, it's not certain. So any probability between 0 and 1, for example, when I say probability 2, this means 20%. So there is a 20% probability that this event can happen, but it's not certain. Of course, the higher the level of, or the higher the figure or the percentage of the probability, the more the likelihood that this event will occur. When you say that an event has, even when you say that this event has a 90% likelihood to occur, this doesn't mean that this event will certainly occur. So probability cannot ensure that a particular event will occur with certainty, okay? But it can just give you an average outcome. This gives you an indication about how possible how the possibility of this event to occur is high. So when you say 90%, so there is a high possibility of this event to occur, but you can never ensure and certify that this event will occur. For example, there is an interesting thing you might have studied, an interesting tool, which is the Punnett square. And you might have studied the inheritance of eye color. Of course, the inheritance of eye color is more complicated than what we have taught you, or what we usually teach our students in the middle school. But we are going to take this example to see or to understand probability. For example, if I say that B capital is the allele for, it is the dominant allele, of course, it is the dominant allele for brown eye color, and that B small is its recessive allele. And by allele here, we mean allelomorphic, or the alternative. So allele here is the abbreviation or the short name for allelomorphic, and allelomorphic means alternative. So we will see this on the diagram. So recessive allele for blue eye color. Let's look here at the two sister chromatids. So if I say, for example, that these are the two loci on the duplicated chromosome, then you have two loci. You have two places for the gene for eye color. In these two places, you might have B capital, you might have B capital, B small, and you might have B small. Okay? All these are alternatives. For example, myself, I might be B capital. My son might be, my elder son might be B capital, B small. And if my younger son is blue-eyed, then he would be B small. Actually, there are two loci for eye color. To simplify, of course, there are more than two loci, but in order to simplify things, we are considering that we have two places, and these places can, or loci on the chromosomes, can be occupied. A locus is a site for a particular gene, a site for a particular, actually, allele on a chromosome. So every duplicated chromosome will have two loci, and you have these possible alleles. Now, if I am brown-eyed, since brown is the dominant, this is my phenotype. This is how I look like. But I might, so brown-eyed, this is a phenotype, okay? But brown-eyed might be brown-eyed because the genotype or the genetic makeup is B capital, or it might be B capital, B small. So I might be B capital, or B capital, B small. Now, if you have two brown-eyed individuals, so you have a brown-eyed that is married to a brown-eyed mom, okay? Now, if I tell you that these two, this couple, or the husband and the wife, have a blue-eyed individual, then this blue-eyed individual is B small. For that baby to be B small, he should have received a B small allele from his mom and a B small allele from his dad. But what about the other allele? Should this other allele, or could this other allele be B small? No, because if it were B small, the dad would be blue-eyed. So it has to be B capital. And for the mom, similarly, because the mom is brown-eyed. So they are brown-eyed, so they should have the dominant allele for brown. But since they had a baby with a recessive allele, or a recessive phenotype, which is the blue eye, then they should have a hidden recessive allele here. So what are the possibilities? If a gamete contains B capital with a gamete contains B capital, then fused together, that contains B capital, fused together, we would have B capital, B small. And then B capital with that one would be B capital, B small. And we have another one, B small, B small. So here we have B small, B small. No, actually, this is incorrect. I'm sorry, I'm gonna erase. Okay, I have to go systematically. So the argument is correct, meaning that when we have a baby who is B, or one of the offspring who is B small, then the mom should have a hidden recessive allele B small, and the dad should have a hidden recessive allele B small. This is the genotype of the parents, okay, or the first generation. Now let's, sorry, this is a P again. So this is P1, the parents, the first generation. Okay, so this is P1. Now let's draw the gametes. Here we have two possibilities for the gametes. We have B capital or B small, and here we have two possibilities for the gametes, B capital and B small. It's obvious that fusion of that gamete with that gamete will result in B small, B small. But fusion of that gamete with that one will result in B capital, and then this one with that one would result in B capital, B small, and this one with that one would result in B capital, B small. So what we do in order not to miss a detail, so we say first with first, and then first with second, and then second with first, and then second with second. So the probability that they get a child who is B small, and having blue eyes is 1 out of 4, because B capital is brown-eyed. And both B capital, B small are also brown-eyed. So the possibility is 1 out of 4 or 25%. This is the likelihood that two brown-eyed parents get a blue-eyed individual. But does this mean that if those this couple or this dad and this mom have 4 children, does this mean that they must have a child with blue eyes? No, not necessarily. They might have all 4 kids having brown eyes. But the more they get of the children, for example, if we suppose that they get 12 kids, for example, there is a greater possibility that you could see 3 out of these 12 blue-eyed. So this means that the larger the sample is, the more the likelihood that the probability can be seen. The probability is 25%, but this does not certify that for every 4 kids, you will definitely have 1 kid with blue eyes. No, you can see this when the sample is large. What I have done here is a genetic diagram, not a Punnett square. If you want to do it in the form of a Punnett square, we would do like this. So we would draw a Punnett square like this, okay? And then here, for example, you have we will divide this into 2. And we would have here, for example, the gametes of the mom, the genotypes of the gamete of the mom. She has 1 gamete that is B capital and 1 gamete that is B small. Here we have B capital and B small. B capital with B capital, okay, and then a B capital with B small, and then B capital with B small, and then this B small with that B small. So again, the B small is 25% of the possibilities. Of course, you are supposed to write the phenotype under the genotype. So genotype B capital is brown. This one is brown, that one is brown, that one is blue. But the difference between this brown and that brown is that this one is pure or hybrid or homozygous, and that one, sorry, this one is pure or homozygous, and that one is hybrid or heterozygous. So both B capital and B capital B small are genotypes for brown eyes, but this individual is considered pure brown-eyed or homozygous, a homozygous individual, whereas that B capital, B small genotype indicates that the individual is not a pure one, he is hybrid or heterozygous. Hybrid or heterozygous, okay? Okay, thank you guys. We will stop here, and in the next session, inshallah, we are going to explain new slides. Thank you.